thank you for joining us today to discuss methylation science and its application in the clinic. What we're going to discuss today is essentially a little bit about methylation and how supplementation, nutrition, and targeted supplementation based on known genetic polymorphisms impact human health. We're not going to cover too in-depth the biochemical implications, but rather we're going to merge the biochemical implications and the clinical application together so that you can understand better how to leverage this into your practice. So when biogenomics is merged with traditional medicine, so the XR-RX approach, doctors can benefit a wide range of illness in a variety of groups, and that includes ADHD, depression, autoimmune disease, and migraine. XRMD's goal is to understand, prevent, and treat based on the cause of disease at the molecular level in conjunction with current methods to ameliorate symptoms. Biogenomic science is the application of biochemistry and genetics to the practice of medicine, and it seeks to support physiology to aid and repair the body's natural healing mechanisms at the molecular level. Biogenomics incorporates bioidentical hormones, methylation science, genetics, vitamins, supplements, chelation therapy when indicated, nutritional recommendations based on biochemistry, and much more. We make biochemistry clinically relevant, which, as you recall from your training leading up to medical school, I'm sure many of you wondered what the practical application of understanding biochemistry, organic chemistry, and biology would be to your clinical practice. And I think now what we're seeing is that we have that capability to leverage that knowledge and understanding of physiology and apply it to medicine. So the diagram that you're looking at before you is very complicated, and we're only going to discuss a small aspect of this today, and that primarily is concerned with the methionine cycle and the related folate cycle. There's obviously a lot of interconnectedness between the methionine cycle and the rest of the methylation pathways. And as you can clearly see, SAMe, for example, which is produced in the methionine cycle, is implicated in a number of biochemical reactions, including the metabolism of catecholamines, and that includes norepinephrine and epinephrine. But we're primarily going to focus just on that upper right-hand quadrant here, where the methionine cycle is involved, and the folate cycle. So I've simplified that diagram and made it into something that's much easier to read and follow. And what we're going to do is we're going to start with methionine, primarily introduced from the diet, but it's also recycled from homocysteine in the body. And we're going to go step by step around the cycle and talk about the clinical significance of each of those biomolecule intermediates, as well as the cofactors and substrates that are essential for not only making the interconversions, but also ensuring that there are satisfactory levels produced endogenously. So the first step in the cycle is the conversion of methionine to S-adenosylmethionine, and we have those highlighted on your cycle that you see before you. That biochemical conversion is catalyzed by an enzyme called methionine adenosyl transferase, and it's abbreviated MAT. ATP is a substrate for this biochemical transformation, where you can see the molecular structures that we have presented at the top of the screen. Essentially what's happening is that methionine has an adenosyl group that's transferred and attached covalently to the sulfur atom. MAT can be inhibited by alcoholic liver disease and some prescription drugs as well. And we'll talk about SAMe in the next slide, which some of you may have heard of or even used in your clinical practice. But this is probably one of the more important biomolecules that we're going to discuss today. So again, SAMe, the functional part of SAMe that actually transfers in methylation, the methyl group, is this carbon with 3 hydrogens attached. And again, that's where the term methylation comes from. It's actually the transfer of a methyl functional group to other biomolecules. SAMe is involved in over 40 biochemical reactions as a methyl donor. It's used in the transmethylation and transsulfuration metabolic pathways, and we'll touch on transsulfuration a bit at the end of the slide presentation today. And one of the interesting things about this conversion, it's not catalyzed by any particular enzyme, although many enzymes are involved in utilizing SAM as a substrate in biochemistry. It is elevated SAW, which is the downstream product of SAMe, does have an impact on how SAMe is utilized in the body. Because SAW is structurally very similar to SAMe, you can see the key difference between the two is that SAW has lost its methyl group once that biochemical reaction has occurred. SAW, because it is structurally similar, can bind to many of the same enzyme pockets and can inhibit SAMe-mediated reactions. So this is clinically relevant. And again, when we get into some case studies at the end of the presentation, we'll actually discuss the significance of elevated SAW. And SAMe is also clinically, you have to be careful when using anything that is a strong methyl donor, as SAMe is, because there can be interactions between SAMe and some antidepressant medications. And there are some reports of serotonin syndrome resulting because of SAMe's involvement in specifically neurotransmitter synthesis. The conversion from SAW to homocysteine is the next step in the cycle. And again, many of you have likely heard of homocysteine. Obviously, it's implicated in cardiovascular disease, elevated levels. This reaction is catalyzed by an enzyme called S-adenosyl homocysteine hydrolase, also known as S-adenosyl homocysteinase. This is a copper-dependent enzyme, and there are actually multiple steps that happen between the conversion of SAW to homocysteine, and two of those reactions are facilitated by NAD and NADH. The byproduct of this reaction is adenosine, and this is important because adenosine is clinically relevant for a number of disorders. Elevated homocysteine can cause SAW levels to elevate as well. So it's important to remember when you're looking at these diagrams and the complexity, there is a lot of feedback, feed forward and feedback interactions. So one substance does not exist in isolation. It's going to impact a large number of other substances in the methylation pathways, and elevated homocysteine is no exception. It can certainly impact the ability of additional SAW to be converted downstream if there's a backlog, if homocysteine levels are elevated. Homocysteine to methionine can happen via one of two pathways. It can actually be facilitated by the conversion catalyzed by betaine homocysteine methyltransferase. And again, this involves trimethylglycine, abbreviated here TMG. The byproduct of that reaction is dimethylglycine, DMG. Again, a methyl group is transferred from TMG to homocysteine to make methionine, so we have a free sulfur functionality on the terminal end of this molecule of homocysteine, which is then methylated again to make methionine. In the second part of the pathway to convert homocysteine to methionine, this is actually where it can get fairly complicated. There are actually three enzymes involved in this process, one of which is methyl tetrahydrofolate reductase, which catalyzes the conversion of folate into 5-methylfolate, which I'm sure many of you have heard of. A second step is actually the methylation of cobalamin, and that's catalyzed by methionine synthase reductase, and that facilitates the transfer of the methyl group from folate to cobalamin to make methylcobalamin. In the final step of this part of the pathway, methionine synthase then catalyzes the transfer of the methyl group from methylcobalamin to homocysteine to again produce methionine. So methionine in the body can be recycled either by the BHMT pathway or by the methionine synthase pathway. Either way will promote that conversion of homocysteine to methionine. There's another pathway in which homocysteine can actually be converted to other essential products that are involved in methylation, and that's down the transsulfuration pathway that I alluded to earlier. And this is catalyzed by an enzyme called CBS, and there are multiple steps after the CBS reaction where eventually one of the desired products is glutathione. And again, clinically, many of you have likely used this in practice. You're certainly aware of it and its significance and its role in human health. And again, this is homocysteine to glutathione. Under normal physiologic conditions, homocysteine, approximately 50% of homocysteine, is converted to glutathione or down the transsulfuration pathway. A CBS single nucleotide polymorphism, which is basically a genetic error that exists within this enzyme system, is going to change how much of that homocysteine is converted down this pathway. So under normal physiologic conditions, if you don't have any genetic errors, approximately 50% of homocysteine is diverted down the transsulfuration pathway. With a single nucleotide polymorphism, this is actually an upregulation, which means that the speed or rate at which homocysteine is converted down the transsulfuration pathway increases dramatically. And something clinically relevant that you may have noticed or been aware of with some of your patients, if you're recommending supplements and or you're noting reactions to certain foods that they're consuming in their diet, is that anything containing sulfur is going to typically cause side effects for people that have the CBS SNP. So if there are supplements such as diacetylendomethane, DIM, MSM, glucosamine sulfate, if they have an adverse reaction to sulfur-containing drugs, or to food substances like garlic, or to sulfites in wine, for example. All of this may be explained by a CBS upregulation. And the reason for this is that you can overload this pathway with excess sulfur. And sulfur, of course, sulfites are metabolized by an enzyme called SUOX, which is beyond the scope of today's talk. But nevertheless, if you overburden that enzyme, often you're going to have sulfite sensitivities that ensue. Additional challenges at this step of the pathway are if you have more polymorphisms than just the CBS polymorphism. So if you have a single nucleotide polymorphism with a gene that codes for BHMT, for the gene that codes for MSR, methionine synthase, or methyl tetrahydrofolate reductase, any combination of polymorphisms in those genes are going to complicate the clinical picture. And so what we're going to do in the upcoming slides is talk about 3 case presentations and how some of these challenges were addressed in the clinic. So the first case study that we have is a 40-year-old chronic migraine patient. She was suffering from migraines for 3 years after the birth of her second child. Migraines were triggered by stress, food allergy, hormonal changes during her menstrual cycle. She was unresponsive to mainstream medical care, and this included bioidentical hormone replacement therapy in an effort to rebalance her hormones and mitigate the changes that were precipitating migraines during her menstrual cycle. And when she came to us, she was having a migraine every time she consumed food. And she was unable to pinpoint what the exact trigger was. She just knew that when she ate, and her diet was restricted down to essentially water and very bland, low allergenicity foods. But she wasn't aware of what in the food was actually causing the problem. So what we did is we proceeded with genetic testing, which allowed us to identify single nucleotide polymorphisms in her methylation pathways. And what we discovered was that she had a double mutation with the methyl tetrahydrofolate reductase gene. She had 4 single mutations, and there are 4 genes that code for betaine homocysteine methyltransferase. She had 4 single mutations in that enzyme pathway. And then the CBS polymorphism, she had a double mutation again, which essentially means that when you're talking about fully functional and you have no polymorphisms, it's going to be working at, provided all the necessary cofactors are present, it's going to be working at maximum efficiency. If you have a single nucleotide polymorphism, the efficiency of that enzymatic system is going to be reduced. If you have a double mutation, it'll be reduced further so that you're very inefficient. It's not an on or off proposition, but rather it's a degree. So you're going to have either highly functional or you're going to have suboptimal. In terms of the methylation testing that we proceeded with for this particular patient, we identified that she had suboptimal levels of glutathione, S-adenosylmethionine, S-adenosyl homocysteine, 5-methyl-THF, and B12. So diagrammatically, when we're looking at this case, what we identified was that at very key points in her methylation pathways, she had significant deficiencies. And again, what we determined, too, from evaluating her case history was that many of the foods that she was reacting to contained sulfur, and they also contained histamine, or were histamine-releasing foods. And this is pretty important at this point because S-adenosylmethionine is involved directly in the intracellular metabolism of histamine via catalysis by histamine methyltransferase. So low SAMe levels are often going to manifest as allergies or sensitivities to foods that either elevate histamine or that actually contain histamine. The next thing that we found was that her CBS upregulation was causing a lot of problems and was manifesting in sensitivity to sulfur-containing foods. So, for example, if she were to eat sulfite-treated preserved fruits, she would get a headache. Again, this was explained by her CBS upregulation. Secondarily, what complicated this picture was that she had the BHMT and MTHFR polymorphisms. These are downregulations. So at the point of homocysteine, when you have downregulations at 2 points that are facilitating the conversion of homocysteine and an upregulation in the CBS pathway, what can end up happening is that you, instead of 50% of your homocysteine being converted down the transulfuration pathway, the percentage actually is much higher. So the clinical recommendations that we had to proceed with first was we had to limit her total sulfur loading in her diet. We had to limit the amount of histamine that she was exposed to. We had to supplement additional molybdenum because, again, SUOX is what's principally involved in metabolizing sulfur and specifically sulfites into less neurotoxic sulfates, which are also water-soluble, so they can be excreted from the body. We then proceeded with supplementation recommendations to overcome the polymorphism with methyl tetrahydrofolate. So she was given methyl THF. She was also given methylcobalamin so that she could work around the inefficiencies that she has in making sufficient levels of 5-methyl tetrahydrofolate. And recall that 5-methyl THF is responsible for methylating cobalamin to make methylcobalamin. We also provided her with supplemental TMG because, again, this enzyme is going to be limited in its function, so it needs additional substrate in order to work optimally well. So we want to open up these pathways, going from homocysteine to methionine through BHMT and also through methionine synthase, and we want to limit the amount of downstream sulfur-containing byproducts and neuroexcitatory molecules that we're actually making. And the end result was, through dietary modification and supplementation on a targeted basis, directly based on her genetic polymorphisms, we were able to essentially rid her of her migraine. So now she is, at this time, hasn't had a migraine in over 4 months. She's able to resume normal exercise routines, which she wasn't able to previously. And notable, she's also taking no bioidentical hormones, where previously it was thought that she was going to have to be on hormones in order to support her monthly cycle. And now she has resumed having a monthly cycle and doesn't have the migraines associated with the changes in hormones. Our next case study is a 51-year-old male that has ankylosing spondylitis. And what's really critical here is that when you're looking at these case studies is to not so much focus on the diagnoses, but rather focus on the biochemical errors and the genetic errors that are causing the manifestation of these classical diagnoses. But what you'll find is that although the diagnosis is different, the underlying root cause is going to be very similar. So this particular individual is a 51-year-old white male, and he has a 13-year history of body aches, fatigue, foggy thinking, depression, and chronic pain. He was diagnosed by a rheumatologist as having ankylosing spondylitis and was told he would require pain medication for the remainder of his life. And much like in the prior case, he had a full workup prior to coming to see us, where his hormones were evaluated, and it was determined that many of them were suboptimal. He, too, was unresponsive to hormone restoration therapy. They didn't improve any of his conditions that he originally had as a result of the ankylosing spondylitis. So when he came to us a few months ago, we ended up doing a genetic test, which are the results of which are still pending. We ordered a whole blood histamine test, and the results came back at double the upper end of the reference range. So the first thing we thought was, okay, this gentleman is going to have suboptimal S-adenosylmethionine levels, because again, going back, S-adenosylmethionine is centrally involved in the metabolism of histamine, intracellular histamine. And his methylation testing revealed that he had suboptimal levels of glutathione, S-adenosylmethionine, SAW, 5-methyltetrahydrofolate, and folinic acid. Now again, looking at the diagram and how it relates to the clinical picture, again, SAMe was low, glutathione was low, B12 was low, methyltetrahydrofolate was low. We don't yet know the genetic results, but we can infer, again, that in all likelihood, he's going to have some difficulty at the methionine synthase reductase, and or methyltetrahydrofolate level. Certainly with the methyltetrahydrofolate gene and enzyme system, his methyltetrahydrofolate levels were extremely low. So again, based on the high histamine loading that he had and the measured levels that were detected, we recommended and proceeded with a low histamine diet. And then secondarily, we sought to address the deficiencies that he has. And 5-methyltetrahydrofolate, as well as folinic acid, which is upstream of 5-methyltetrahydrofolate, methyl B12, and methionine were all indicated. Now, in some cases, and in this particular case specifically, he did not respond well previously to S-adenosylmethionine supplementation directly. And if you're looking at the order of magnitude for a lot of these supplements, S-adenosylmethionine is often given in 200 milligram dose increments. Methyltetrahydrofolate is often on the order of microgram or milligram scale, as well as B12. So S-adenosylmethionine, in terms of its capacity as a methyl donor, is several orders of magnitude more potent than either 5-methyl-THF or B12. They all serve unique functions in the methylation pathways, but in some instances, S-adenosylmethionine can be overstimulating for some people if given too early in the treatment process. And again, for glutathione, often what we end up doing is treating the rest of the cycle and the blocks and or deficiencies that exist, and we find that glutathione levels actually increase. This particular individual has now been able to titrate off of most of his pain medication. His diet is improved, his mood is improved, his energy is improved. And while we don't yet know what additional polymorphisms might exist from his genetic profile, he certainly has made significant improvement in his overall health and his ability to function simply by evaluating his methylation pathway analysis. And we expect that we'll be able to take him to another level once his genetic results are known. The third case study is another chronic migraine sufferer. This is a 36-year-old white female suffering from migraine since her mid-20s. Migraines are triggered, in her case, by environmental and food allergies, hormonal changes during the menstrual cycle, which is much like the first case that we presented, and she was unresponsive again to mainstream medical care. So she had tried a number of prescriptive and hormones, as well as abortive and prophylactic migraine medications, none of which helped her tremendously. So she was also using acute onset pain management through opioid narcotics in order to manage her pain. Her genetic testing revealed the following SNPs. So she had a double mutation in methyltetrahydrofolate reductase. She had two double mutations in MTRR, which is the gene that codes for the enzyme methionine synthase reductase. She had three polymorphisms for BHMT. There were single mutations. And she had a CBS single mutation. Her methylation testing revealed suboptimal levels of glutathione, SAMe. In this case, her SAW was elevated, which is notable because, again, going back to the early part of the presentation when we were talking about S-adenosylmethionine and SAW being elevated can block the effects of SAMe. So even if SAMe is in range and you have elevated SAW, you may not be receiving the full clinical benefits, whether it be from supplementation or the levels that you're making endogenously because of that SAW elevation. 5-methyltetrahydrofolate she was low in, and her adenosine was elevated. And again, we discussed in an earlier slide where adenosine is clinically significant because of its role in vasodilation. And of course, her B12 was also low. So diagrammatically, looking at where her errors were and some of the challenges existed. Again, the SAW levels being elevated impact SAMe. So SAMe was low. This is further exacerbated by elevated SAW because of its ability to bind to some of the same enzyme pockets. Adenosine was elevated, which is the byproduct of the SAW to homocysteine conversion. She again had an upregulation in the CBS polymorphism. Her glutathione again was low, and again, the low glutathione is likely going to be attributable to oxidative stress and the negative reinforcement cycle that exists once these methylation pathways get disrupted. Her BHMT enzyme pathway was blocked. Her MSR enzyme pathway was blocked, so she wasn't able to efficiently methylate cobalamin to make methylcobalamin for use by methionine synthase, again, to convert homocysteine to methionine. And her methyltetrahydrofolate reductase polymorphism limited the amount of methyltetrahydrofolate that she could make. It was determined by working through and with her primary care physician that she actually had lead toxicity in addition to some other heavy metals, but lead was most prominently involved. And the reason why it's illustrated here is that lead also inhibits the activity of methionine synthase reductase. So not only did she have a polymorphism with the enzyme methionine synthase reductase, or the gene that codes for the enzyme, she also had lead. So she was doubly affected in this sense, where she was very limited in her capacity to remethylate B12. So the first thing that was initiated was metal chelation in order to lower her body loading of lead. And after she had completed about 5 of the 10 chelation treatments, her migraine started to improve. So on chelation alone, her symptoms and her primary complaint of migraine was improved significantly. The other thing that we proceeded with was recommending a low-sulfur diet, again because of her CBS upregulation, and something that we're not discussing today, but again as part of the methylation pathway, and I mentioned it earlier, is her SUOX enzyme. She also had a polymorphism in SUOX. So she's going to have a higher requirement for molybdenum in order to metabolize a lot of the sulfur that's produced down this pathway. Secondarily, much like the other cases, we recommended supplementation with 5-methyl tetrahydrofolate. Again, methyl B12, not only because of her enzymatic deficiency, but also because we needed to get this pathway up and running again. Trimethylglycine, again, to help at the secondary pathway, the conversion of homocysteine down what's called the short path to methionine. And then again, methionine rather than S-adenosyl methionine in order to elevate SAMe levels. And then, of course, other support nutrients as well that are not necessarily related to this part of the methylation pathways. So what we did today was to discuss the methionine cycle, some of the enzymes involved, some of the biomolecules involved, principally SAMe, SAW, homocysteine, methionine, and then supplements that were involved in facilitating the conversion from methionine to SAMe, SAMe to SAW, or rather SAW to homocysteine, homocysteine back to methionine via one of 2 pathways, the BHMT or the methionine synthase pathway. We also talked about some of the complications related to the CBS polymorphism, which is an upregulation again, and the key clinical indicators that you'll be looking for in the absence of having that detail are going to be sensitivity to sulfur and sulfur-containing foods. Now, something else that's important, again, is to keep in mind that the reason we focused on methionine today and not the entire pathway that you see before you is because many of the biomolecules that are made, and principally SAMe again, are involved in so many other aspects of your physiology. And do help determine whether it be depression, anxiety, migraine, autoimmune conditions, the extent to which someone is going to be resistant to conventional treatment, and also may explain why they have a history of various illnesses that they do. So the inclusion criteria for what we're talking about for methylation pathway science. Primarily we're talking about the conditions that are listed in front of you, but it's certainly not a all-encompassing list. There are a number of other variations in the classes of disorders that are listed here, but certainly these are ones that we see most often in the clinic that we have success with in using methylation science. So we can treat migraine, fibromyalgia, ADHD, chronic fatigue, lupus, Hashimoto's, and the list goes on. The important thing to remember is by knowing one's genetics, and often what we end up doing is treating families, you can intervene on behalf of an individual's children. So for example, if often what we see is that a patient may have fibromyalgia or chronic migraines, they have known defects in their methylation pathways and in the genes that code for those enzymes in the methylation pathways. And often what's going to happen is that their children may show early manifestation of those physiologic errors in the form of ADHD, autism, or other developmental challenges. And this is often explained by and revealed by their parents' illness. So through nutritional intervention, targeted supplementation, you can certainly correct and overcome those errors that exist. Thank you very much for your time today, and a little bit more about XRMD, and you may have a question, how do I implement methylation science into my practice? Again, because this can be very complicated, and today we only focused on one aspect of methylation science, and that's related to the methionine cycle, and secondarily the folate cycle. What do? XRMD supports you and your practice with the exciting field of biogenomics. There are no upfront costs. There's no inventory involved. There's real-time patient support. So this is the didactic process. We go through the patient care process with you and support you in the consultancy efforts to help your patients overcome methylation and genetic defects. It's a minimal burden on your time, so you don't have to worry about going back and reading all of your biochemistry textbooks. You actually learn as you're going. You increase practice revenues and you improve patient outcomes. And certainly, as I alluded to, when traditional medical therapies and bioidentical hormones alone are not sufficient to address your patient's health concerns, often the root cause of those disease states are going to be explained by genetics and methylation defects. And then when XR and RX merge, you and your practice and the patient's health wins. If you're interested in learning more about what XRMD does, you can certainly visit our website at xrmd.com/MD.